Welcome to NewYorkBIO's Virtual Breakfast Series, a digital program created in 2020, bringing you fireside chats with leaders from across the healthcare spectrum. This week's episode features Barry Kappel, founder and CEO of Sapience Therapeutics.
All right, I have 9:01, so let's get started this morning. My name is Jennifer Hawks Bland, and I'm the CEO at NewYorkBIO. We're very happy that you're joining us for another edition of our virtual breakfast series. This morning, our guest, Dr. Barry Kappel, the CEO of Sapience, is going to talk all things early stage, founding a company, and some pretty amazing results that they're already getting. So we're really excited to delve in with him. But first, we couldn't do our weekly breakfast discussion with our distinguished guest without the support of our sponsors. So special thanks to this month's sponsor, law firm McDermott, Will & Emery. We're very glad to be partnering with them. And as always, my housekeeping reminders for you, please ask your questions of Barry in either the chat or the Q&A box, and Derek and I will get to them as we go through our conversation with Barry this morning. With that, I'll turn it over to Derek to kick us off. Thanks, everybody.
All right, good morning. Fall is in the air. Barry, how are you this morning? It is great to have you on with us.
I'm doing great. It's great to be here. Thanks for having me.
All right. Well, we tend to kick these things off with a bit of an origin story, and one of the things that I've been looking forward to for a week is you have absolutely got just a wonderful New York entrepreneur origin story. So, do you want to walk us through a little bit of really kind of how you came to New York and how you got to where you are?
Sure. Well, it starts very easily. I was born in New York, so that's the easy part of the origin story.
No, you got to talk longer. We have an hour, Barry.
All right. We have an hour. Come on.
I think origin story is really interesting. I love this program. I've been watching y'all for over a year now, and it's really great to hear the different origin stories of how people arrive in New York. And I said, I started in New York. I ventured out for a little while for college, but made my way back. And it's always been a goal of mine to help not only build companies, but help make sure we have an industry here. And I think looking backwards from today, you can see a straight line from where I started to here. But where you look where we started back in high school or college, there were a million paths, and this wasn't even identifiable, right?
Yeah.
I was always a science, math kid, growing up, and to me that meant you're a physician. That's what I thought you did if you were good in math and science. So I went to Emory University, studied chemistry there, largely because after taking chemistry, there were the fewest number of classes I needed for my pre-med requirement. And that was my goal, was all about getting to med school.
Barry, I have, in your honor for Emory, I have my Atlanta mug I'm drinking coffee from today.
There we go. But while at Emory, I had an opportunity to do some work over one of the summers volunteering at a hospital, shadowing some doctors. And that was, Derek, there are always those formative moments, right? And this was one of them, because my first time in the hospital, in the emergency room, they're doing a surgery, and next thing I know, I'm on the floor being woken up, and I'm like, oh. All right, so maybe physician's not the right. So what can I do differently? I had to sort of think about that first level of pivot. I still took the MCATs and did all that, but I started working. I spent a year doing neuroscience.
Radiology was still available.
I spent a year doing some neuroscience research just to really get some research under my belt and hang out down in Atlanta. I worked at Georgia State University, Dr. Alber's lab. And then I came up to New York and found myself a job at Memorial Sloan Kettering Cancer Center. It was a great opportunity. It was really like building a biotech, because I was the first person hired by a new postdoc from the Dana-Farber named Marcel Vandenbrink. His first week on the job, we literally drove back to Boston and trained on techniques of doing transplantation with his former tech, so the tech could teach me. We came down, built the lab. Within a year, we had 11 people in the lab. We were in a bigger lab. He became the head of hematology oncology, which he still is, and has bred a whole amazing number of scientists, right? And that was great for me to see science. And from there, I decided to pursue a PhD at Weill Cornell Sloan Kettering, with the goal of specifically working in David Scheinberg's lab. I wanted to work on translational medicine and science, and it worked out because he was taking a couple of students that year, and I got to go work with David. I always thought I'd be a professor still at that point, though. That was sort of my goal. It was a really great opportunity to see translational medicine. Jed Wolchek was my next door neighbor on the floor. Jim Allison's on the floor above us. I was working with Michelle Setterley and Munir Brankins. You're seeing immune oncology come to be. Translational science is happening. It's wonderful. And then, NIH funding in the early 2000s was really tough for first-time professors. So as I was finishing up school, I got to talking with Dr. Dean Hajar of Weill Cornell Medical School. And he's asked me what my plans were for the future. And this was in early May of 2006. And he said, well, have you ever thought of business school? Because I was talking about postdocs and really not being thrilled with looking at, $25,000, $30,000 a year jobs after graduating from Sloan Kettering, trying to start a family. And, he went out of his way for me. And the next day, the dean of Cornell Business School called me up and said, we know the semester starts in 2 weeks, but if you'd like to come to our business, we'd like to have you if you could take the GMATs tomorrow. So I did. And then I rented a car. My mom drove, actually, I didn't rent the car at that point. My mom drove me up to Ithaca the next day and I wrote my application essays on the car ride there. Interviewed that day, was in the next day, bought a car the next day, drove to Ithaca, rented an apartment and started a business. My fiance is still at home. So my car, for a year, I went back and forth to Ithaca. But it was a great program. It was called the 12-month option. It was literally from Memorial Day to Memorial Day. And while you're there on campus, you really get to make that transition from academia to business. They come recruit you on campus. And my goal was to fund or found science-driven companies when I went there. And I met some folks from a small boutique life science consulting firm, Easton Associates in Manhattan. And I never wanted to be a consultant really because I didn't know what that meant. But when I heard them talk, I wanted to do what they did. And that was, I felt I can learn the most about our industry in the shortest amount of time and really have fun while doing it. And that was a special place and special people over there. I mean, the 40 people I worked with over there, I can name every one of them. I probably know what they're doing. They've spread out, whether it's staying in consulting and running their own consulting firms, moving into BD positions. It was truly a wonderful 2-year experience. I recommend anybody who wants to leave academic science and get into business to go through life science boutique consulting. Mm-hmm. And then from there,
it's funny. I know a bunch of people from your class-ish thing. That group was awesome. There are so many people that happen to work together during at Easton during that time. It's a really great group.
It was amazing, and we're all still in touch. And I missed JP Morgan. We'll get the alumni event last year, and don't know what happened this year, but they really are special folks. And then in 2009, that's where the journey really takes a turn towards entrepreneurship. I was leaving consulting and had opportunities at Big Pharma or to join Contrafect as the first hired employee. It was Bob Nowinski had started the company, a serial entrepreneur. I had a 6-week-old daughter at home, and I'm like, well, if I'm ever going to do it, now's the time. I don't know if that was the right choice or not. Figured if I'm there for 2 years, it's a decent experience. If I'm there for less than that, I could still get another job, and then more than that, it's great. Well,
I almost want to have Lucy on for the second half of this show and ask her all of the same questions and have her standpoint on all of these stories.
Well, let's see. We came through the same lab together, went to the same grad school together, and she comes back into the story shortly when we start Sapience. But that's my wife, who you can see in that little picture in the corner over there. But that's about the time we joined.
For the edification of the audience, Barry's wife is Lucy Kappel, who's in BD at Regeneron and is also one of the smartest people you will meet in the New York area biopharma scene, hands down.
No argument for me on that. I'm thrilled if she's the better half of this relationship. That makes me exceedingly happy.
That's not on my topic sheet. I don't know if we can cover that today.
But it was a great opportunity to join and start a company and be number 2 at a company with someone, 30 years my senior. I found myself constantly in the room, being 20, 30 years younger than the folks around the table and just learning, absorbing, hearing what it was like in the 80s and building a company from, 2 employees to 30, building an entire laboratory facility, raising the first $1 million, raising our 100th million on our NASDAQ IPO, licensing assets from the Fischetti lab at Rockefeller that are currently in, I'm guessing, late stage phase 3 studies for important indications. It was an amazing experience. But in 2015, it was time to sort of go out and get back to the purpose of what we wanted to do, and that's build an oncology company. And that's where Sapience came from at that point.
So how did you decide? So you had sort of set a path, right, at Contrafect, early stage. Was it being there and seeing all that built that gave you sort of the desire to do it yourself, though, to sort of control the process?
My last 6 months at Easton, one of my friends from business school who was there with me, John Parrish, he and I were actually talking about maybe starting our own company. And we went and spoke to some of my former colleagues like Jeannie Farrell, who was at the time, I think, still at Sloan or Mount Sinai, now at Rockefeller, and that was from our Cornell days. Just looking at assets around the New York area, see if there was something we can build. But then Contrafect happened, and I thought it was a smarter way for me to learn what to do.
Right.
I don't think that Sapience would be nearly as successful as we had been if I didn't get to be a little bit of a witness and the person, doing the work, so I wasn't necessarily setting all the strategy and tactics early on, but I was executing them.
Yeah.
But the exposure at Contravec was great because I really was like the consigliere of the company, right? I was there in all the meetings, got to execute the plans, got to do some hiring, got to run preclinical, got to do some finance work, and got to meet people, meet the board, present to them, meet the bankers.
Yeah.
I mean, that was key. I mean, when I left in 2015, I had board members and bankers who said, we want to do what you do next, we'll be with you, right? So it gave me the confidence to go out. And I didn't go out without a mission. When I left in 2015, I gave myself a certain amount of runway to find assets. I gave myself criteria to find those assets. And I went out on a mission to do it, and got it done.
That's really smart. And I think your Contravec experience is something that people kind of tend to overlook when they think about starting a company. A lot of times I think we have a bit of a romantic notion about company formation, which in some senses is true, but there's a lot more that goes into it that I think people don't appreciate. There's a lot of things that are difficult that people don't appreciate. And actually, being able to go through it once when you don't have, 100% of it on your shoulders is actually really important. So what are some of kind of the big things from Contravec that you were able to kind of take away, whether it was culture, whether it was kind of formative experiences, right? So what were some of the big things that you were able to take away from that? Good or bad too, because there's things you see about running a company where you say, if I'm going to be able to do it next time, I may want to do it differently.
I would say, first of all, probably equal parts good and bad that I learned lesson-wise that I wanted to take forward. I learned a lot about myself. I learned that probably my favorite part of the job was making sure all the employees we hired had food on the table, good healthcare, loved coming to work every day. The things you don't think about when you're the scientist starting a company initially, right?
Yeah.
I love getting into the finance side of the work, meeting with the bankers, meeting with investors, doing the modeling, strategizing those aspects of the work. I thought we did some things wrong there. I thought we built big early. That was Bob's vision, our former founding CEO. We went from a company of 2, we built out lab that could house 30 to 40, right? And 50 on our Series A B proceeds, right? Not on, real big crossover proceeds. So we took a lot more deliberate approach here at Sapience. We've been 5 years and only have, today we're 6 years old, by the way.
Yay.
Yeah. Congratulations.
We have 12 employees, but we've accomplished a lot more than I think most folks can in that time because we're efficient and deliberate. So that was something I really wanted to instill here. But we are a people-first organization at heart. Every hire is important to us. Everyone fits in well. Most of the folks I've worked with, at least the initial team I've worked with for between 5 and 25 years. So I said, Contravec was a great experience, but I learned from being up in executive levels too that, you really gotta, you say something, people hear it, and you gotta follow through. Things that you think might be simple things are important to everybody, and really just be a man of your word, both to your investors, your people, your employees, and just.
And how many, at the beginning of the pandemic, how many employees did you have out of your 12?
We had 9 at that point.
Okay. So yeah. So, and then you still increased over, even as people were remote. And I know you all have been back in the office earlier than a lot, just because of your lab work.
So the increase really came the second half of this year, more so. But yeah, we shut down the labs for around 5 or 6 weeks last year, totally. One of our folks who lives locally came in and just kept some of the studies that were ongoing.
Yeah.
But within a couple of months, we were back to running, shifts in the lab. And I've been back in the office personally because we have our own office since June of 2020. But everyone still, comes and goes as they need. But because we've worked together for so many years, because everyone on the business side of this company, which at that time was only one of us, but 2 of us, and is now 3 or 4 of us, can trust each other. And the head of research, Jim Rotolo, I've been with since 1996 at Emory, right? 1995. We were seamless during COVID.
Yeah.
If I showed you timelines in 2019, they're the same timelines I could show you today. And that means regulatory filings, clinical trial starts, clinical trial enrollment, second program development, closing financing, hiring, we kept the whole chain moving exactly as described. Folks who saw us at January of 2020 JP Morgan would not have known that COVID happened when you looked at our company. And that's a testament to our employees and their work ethic throughout it.
That is so rare. I mean, it never happens. You know, it's one of those things where you put the plan together knowing full well that something is going to change. But that's pretty remarkable that you were able to do that. And it's funny, you know, in thinking back on the contrafect stuff, it's interesting because hindsight is always 2020, right? And you have kind of the luxury of kind of seeing how decisions worked out, and then kind of remember like the process of how the decision was working out, and the end result just kind of sticks in your head. But you know, I think one of the most important things that you said is that you learned a lot about yourself, and you learned about the stuff that you were good at and the stuff probably that you didn't like quite as much. So I imagine that kind of helped you as you were building out Sapience to, you know, generally understand, okay, this is how I want to build things. This is where I need either people or other structures around me. This is the stuff that I'm going to dive into. This is the stuff that I'm going to have to force myself to do because frankly, I don't like it very much.
For sure. You know, I think that's a good point, Eric. You know, so some of those things about myself and about the organization, I think that take from contrafect still ring true through the early years of Sapience and now. You know, we're not a meeting heavy organization. You know, we want people to work. I like to hire people and empower them to do their jobs. You know, I like to almost end every meeting saying, what can I do in the next week to make sure you have the opportunity to do your job successfully, right? Those aren't words we heard at contrafect. It was, you know, that wasn't the tenor of management early on. And not to say what it's like now. I honestly haven't been really inside the walls of there in 5+ years. They've continued to grow and do great stuff. So this is, you know, I have no ill will. I actually love this one. You know, my first baby, right? Contrafect. I love that.
Yeah, I know.
But at Sapience, it's just a different organization that we built. You know, so really we're science heavy early on. You know, it was just me and an assistant on the business side until the summer of 2020 when I brought in my, you know, Nancy Dong, who was from contrafect, came over as my SVP of finance and admin. So we've taken a lot on our shoulders. We do a lot. We ask a lot of our people. We work with great consultants, you know, which help us do it as well. Keep it efficient for us.
So if we wind back to where, you know, Sapience hasn't started yet, and you actually are thinking about literally, you know, what to do and how to put things together. So what was your process like early on? What were the things that you genuinely wanted? What were the things that kind of, you know, shook out in the weeds when you were able to find assets? What was your initial vision that you wanted to build around?
So I'd say that probably the best thing that happened to me when I left contrafect was the next day, Isaac Bleck and Chris Brothers. So one of my board members and Chris, who's a banker at the Maxim Group, who really is like an angel for our industry here in New York. I mean, his group has raised me $136 million for my companies over the past 10 years. Great with the private financings for early stage biotech. Those two took me out for lunch and said, if you find something you want to build a company around, I will be a board member and I will help finance it. And I went out with confidence. Confidence is obviously important in this game. I then had a set of investors who were interested in looking at something in Israel and asked me if I'd fly to Israel with them. This is all within 24 hours of leaving contrafect. So I was on a plane, you know, less than a week later to Israel. And that was a great experience. I didn't like the assets there. But when I came back to the United States 5 days later, it was like I was gone for a year. Contrafect was in my past. And then I was able to have clarity. And I was able to define exactly what I was looking for. I went out on a mission to find academic assets to build a company around that could had three qualities. One, despite having a degree in chemistry, I wanted a peptide or a protein. That's what I've worked on. That's what I know. Those are the kind of drugs I wanted to develop. I thought they were similar. I was very wrong. Peptides are very different. And I'm a big fan of that, now that I know what they are. Number two, I want to say that had a good amount of data behind it. Something I thought could be 18 to 24 months of IND if the academic work holds up, you know. And with our molecules, some did, some didn't. I think with academia, we know that's the case. But you hope you have a good hit molecule, and we did. And third, I wanted something to address an important unmet need. And that could be defined any different way, right? For us, it was diseases that, despite standard of care, had greater than 50% mortality. Mm-hmm. So with those criteria, I started just going to my network, looking at LinkedIn, going to all the tech transfer offices, making calls, meeting people. I spoke with everyone from, you know, Cold Spring Harbor and Stony Brook to UCSF, right? And everywhere in between. And it turns out, and probably looked at 200, 300, 400 programs over a 3-month period, right?
Yeah.
Having a wife who's an oncology BD professional at home is very helpful.
Yep.
Like if the two of us could find something we like, it's probably something I can rally the troops around and get investors interested in as well. Turns out that right at Columbia University, 5 miles from my house, they had a program that fit all the criteria. In the lab of Lloyd Green, he was working on peptides that can be delivered systemically, could travel around the body, could cross the blood-brain barrier, can get into the cells, get into the cytoplasm, into the nucleus, but could bind to and disrupt a transcription complex that was responsible for an oncogenic event in glioblastoma, the most severe form of brain cancer. So that was, peptides, data, glioblastoma, fit my criteria. Found that program in July of 2015, so within 3 months of the search. By October of 15, we had a term sheet in place and option license agreement signed, incorporated the company, and we're kind of off to the races at that point.
There you go.
That's it. How did you decide on the name?
That was harder than you would think. It was I used to spend a lot of I was very much your stereotypical entrepreneur, especially during the summer of that first summer. I would go to the coffee shop in the morning and work. And then I'd meet a friend who was also in between jobs, and we'd play golf at the local community courses in Westchester. It was a great, very fun summer, very formative summer. And I spent a lot of times, when I was at the coffee shop and tired of writing a business plan, I would just think about names and think about what I wanted and couldn't find anything that really fit. And then if I came across the word Sapience, and I think Derek tried to correct me early on, telling me I pronounce it wrong. It should be Sapience, but I still call it Sapience.
Did I really?
I think during our lunch outside of on the Upper West Side somewhere, but it's a word.
Oh, no, I honestly, I hadn't heard that word before. I don't think I would be. I don't think I would be so bad to try and tell you were mispronouncing something. I'm bad, but I'm not that bad.
But it means knowledge. It means thoughtfulness. It means wisdom. And we thought that was a great word, a great meaning to start a company behind, because that's what we wanted to do. We were coming at oncology with new approaches, which I think biotech should do. In 2015, we could have started another pure play immuno-oncology company. With my background, it probably would have made sense, but it didn't feel inspiring to find another academic PD-1 molecule at that point. So we wanted to do something different and thoughtful, and we really wanted an approach to go after what's called undruggable targets, and so I think it fit very well.
It's funny. I remember that really, really well. And for the edification of the audience, we were outside at a place called Deluxe, which is down the street from Columbia. My son was 6, and as far as I remember, he behaved pretty well while we were sitting there having a beer. And I remember talking about the name. I remember the name at Knowledge. I remember you telling me about the peptide program. I remember you telling me about glioblastoma, and I remember being really excited for you. And this is one of the things that happens with new entrepreneurs or not. I think everybody gets genuinely pretty excited for them. But I think the reason behind that was that all of the stuff that you were talking about was really well thought out and was there weren't any holes, right? It's not like you were jumping into the void and just super excited about it. I think the reason that I was really excited for you was that you seem to have a lot of these things in place. And I think the great thing now is that, it turns out that you are, either right on a lot of it or that a lot of it's come through, right? It's really easy to feel good for somebody that's starting a company because you want them to be successful. And the vision of the company that you get in the first meeting over coffee is probably 30 seconds of detail. And it's rare that, all of that plays out the way that you want it to. But I remember vividly being super excited for you because you had this depth and really, quite honestly, because it wasn't just another PD-1 play or anything like that. You really had something that was a different angle and on something important. And really going into something like glioblastoma, it's super difficult, but those are the patients that kind of need it the most. And it's one of the things that we're excited that you did, because if people don't do the things that are hard, you never make any traction against the things that are difficult, right? So, I mean, you harken back to you look at PD-1 and you look at ipilimumab, And if they didn't go into, stage 4 melanoma, we would still have a disease that was, a 10% survival rate at 5 years. So I remember that. I don't remember you. I don't remember dinging you about the pronunciation of the name, but
Oh,
I
you
do
totally
remember
did.
the rest of it. I don't think I do. I'm not. No. Honestly, I'm a terrible Scrabble player. I'm actually. I do not have the world's largest vocab, and I don't. I'm pretty. I don't know. I don't know
It
that
could have been
stuff
someone
very
else
well.
around the same time, but
It's very. It's. I don't think that would be me. I could give you a hard time about a bunch of things, but word pronunciations, especially ones that I don't know, I think that's above my pay grade. So we have a question from the audience about your webpage has something that says you're searching for new partners, and why don't we talk a little bit about just the way that you've kind of developed your pipeline, right? And then maybe go into how you think about BD from this stage of the company.
now, and that's actually a perfect segue, because everything hasn't been smooth sailing, right, the whole way through.
Yeah.
The idea is to be able to get through the bumps, without people seeing them, have enough money to,
Yep.
and that, your inside folks know what's going on, your board knows what's going on. But if you have the right plan in place and you thought the financial strategy appropriately and are using your capital efficiently, you get through those. And look, we had an early moment, and this leads into how we developed our pipeline. We licensed the assets actually in April of 2016 when we closed our financing. And at that point, I brought on my first scientist in June of 2016. And we started outsourcing some work, right? We weren't going to necessarily build a lab right away. And we started working with some CROs, and we were sending out molecules, and they weren't performing as expected, right? Mm-hmm. So early on, when we went back to the board, we're like, we've been through a CRO. We thought they messed up, went through a different CRO. We thought they messed up. We got a lab bench. We don't think we messed up. We don't think all three of us are messing up, or something's not, these aren't recapitulating exactly as we wanted them to. And we had that moment early on, sort of, well, we still have $20+ million of our 22 in the bank. Do we abandon ship or reinvest and try and figure this out? And we said, let's take it sort of in 3-month increments. And the first 3 months, we did something to the molecule that we thought made sense, and we boosted activity 15-fold. And then we spent another 3 to 6 months sort of understanding that change, and then SARing the molecule. So doing some structure activity relationship, bringing on our peptide chemist, Gene Marutka, who is one of probably the world's foremost peptide drug developers, to really help build out that first rational library off of that molecule. And by the time we got to December of 2017, we had ST-101, right? It was a great process, but along that road, it was a little difficult. So what we figured is we could take academic molecules that are hits, not pharmaceutical leads, and could probably do this again. So what we did is we went into the literature and found other peptides that we didn't have to license at this point, that we could look at. And we said, okay, why don't we do this again? And we did. And that's ST-316, our second program. But then we said, okay, we could be a company that has cherry picks things from literature, but if we want to be a true biotech discovery company, we need a discovery engine. So we went into the literature, and we couldn't find any companies that were doing peptide discovery work like we are. There are companies that do peptide discovery work, but not big therapeutic peptides like ours, right?
Okay.
So what we did is I found a professor at the University of Bath who was running a really cool assay that could discover peptides like ours. The University of Bath in England. I reached out to him. He's now on our scientific advisory board, Dr. Jody Mason. We have 2 full-time postdocs that sit in his lab and do drug discovery for us. We own and operate the drug discovery platform here. We brought down a senior scientist from Genosha Biosciences from the Boston area, come out and build out our platform here and push the technology even forward to discover new types of peptides that we want to work on in the future. So we complemented it with a discovery engine that could discover the hits on their own. So now we have sources of hits from literature, sources of hits from our library screen system, and they feed into our rational design stage. And that's where building up the lab was really important, right? We have our own peptide synthesizers. We have out of our 12 people, 9, 7 are full-time in the lab, 2 more are our heads of our groups that spend time in the lab, and only 3 of us really sit on the business side, right? And that's because the platform, the pipeline, that's the lifeblood of the company.
Yeah.
I'm sorry. So as we said, so as ST-101 has moved to the clinic and developed that sort of proof of concept data for us, remember, it's not only our lead, but it's a prototype, because nothing that looks like a Sapience peptide has ever been in people before. So it needed to show that it was a pharmaceutical agent, and now it has, right? It's got the right half-life, it's non-immunogenic, it's non-toxic, it gets into tumors, we show PD changes, we're showing responses in patients. So now pharma and biotech companies see the clinical data from our lead program. They see a whole host of very interesting targets in our pipeline, including probably our second program, ST316, which goes against the most important and targeted of undruggable targets, beta-catenin in the Wnt pathway. You know, they can see the two data sets together and see the future of the second program in the pipeline. And a lot of BD opportunities have started to emerge around the pipeline as a result of ST101 success.
We'll get to that, but before we do, I mean, you mentioned a couple of times that yours is a little bit different. So what scientifically is different about your molecules and your platform, if you can tell us?
So all of our molecules, all of our peptides, so okay, let's start basic. Peptides are built from amino acids, so are proteins. If you keep it below 60 amino acids, it's likely considered a peptide. Above 60 amino acids, likely considered a protein. The big difference is probably the rigidity of the structure. Once you start getting secondary, tertiary structures, really tertiary structures where the proteins are really tightly folded, they're proteins. Peptides tend to be floppier and can be different conformations and different equilibriums. So there are different ways to build peptides. We wanted to keep our peptides to a certain length, so small enough, so we could build them by solid phase synthesis, so by chemistry, and not have to use recombinant means and grow them in bacteria.
Yeah.
One thing is we keep them small. We keep them under 40 amino acids in size. We also we needed to stabilize them. Right? So when I would go talk to folks, you know, like Carlo at Versant or other folks, you know, around the neighborhood, when we were starting out the company with the technology from Columbia, they said, you're going to run into the typical peptide problems, right? They're going to be digested by proteases. Digested by proteases, you're going to have no exposure. You are going to be possibly immunogenic. You're likely going to be toxic. You're not going to be effective. So the first thing we have to do is stabilize.
So in layman's terms, if it's digested by proteases, basically the body is eating your drug before it does anything.
Correct. Exactly.
Yeah.
And that's what our body does to foreign proteins and peptides.
Yep.
It surveys the body for them. It breaks them up. It actually puts them on the surface of your cells to expose them to the immune system to say, do we have to mount an immune response?
Right. Get rid of this. Get rid of this. Yeah.
So our first job was, how do we stabilize these? And a lot of folks have different tricks. You can put in salt bridges. You can put in hydrocarbon staples. Company like Bicycle Therapeutics, who use peptides, they put in 2 cyclic rings and they call them bicycles, right?
Yeah.
We wanted to keep it simpler. We want to go to the biology. What does our body build peptides and proteins from, and how do we digest them? Well, everything in nature has chemistry, right? And it could turn left or turn right, right? So like our hands, they're not superimposable, they're mirror images of one another. Everything in chemistry is as well, right? You come out of a bond, you could turn left or turn right. Our body uses something called L-amino acids to build peptides. Those are what mammals use. So we figured there's an enantiomer to it. There's an opposite, a mirror image called a D-amino acid. And by using D-amino acid substitutions of naturally occurring amino acids, we felt we could build our molecules because the way we use them and retain full activity without being sensitive to proteases anymore. And that hypothesis has played out to be true. So by no longer being break by proteases, we have long half-life. So we have a once-weekly administration of a peptide in the clinic, right? We're non-immunogenic because not only do we keep them short enough, we keep them long enough. If you keep them above 25 to 30 amino acids, and they're not digested, they should be too big to be loaded into those immune compartments to mount an immune response. That was a hypothesis, but we've never seen an antibody form in an animal or a human. And we've been in patients for over a year, right?
That's really interesting. So you're in this kind of sweet spot window.
Yeah, and it has to do with the way we use them. You know, they wouldn't work probably as enzyme replacements, but our molecules tend to be like, they look like springs or alpha helices, right? And it doesn't matter for us if the coil turns left or turns right. It's what are the two faces of the helixes do. One interacts with the target and the other provides intra and intermolecular properties, manufacturing properties, solubility, stability. And we get that regardless of D or L isomers. So that was our approach to stabilize these molecules.
That's interesting. The chirality trick is something that George Church talked about in his book, basically about creating these entire, D-based organism things. So it's actually really interesting to pull kind of an evolutionary trick on the body like that. There was a question that asked, CAR Ts were popular at the time that you founded the company. Did you always want to do this, or did you ever think about going to CAR Ts? Because honestly, the wave of the wave of cell therapy companies in the middle of the 2010s was huge, right? Everybody was following Juno and Kite. It was tons of money thrown in that space. Did you ever almost get caught up in that, or did you really have kind of the target in mind and the kind of things you wanted to do with peptides?
Cell therapy is also one of my backgrounds. And like I mentioned earlier, I got to work with Michelle and Renier, who are some of the scientific founders behind Juno, and in David Scheinberg's lab as well, right? That they do a lot of that work.
Yeah.
I love that work. I always thought there were challenges with it that I didn't know that we'd overcome. The manufacturing piece of it, I always thought would be a struggle. I didn't know how we'd commercialize those. And to be fair, those opportunities were bigger than me at the time. Barry Kappel wasn't getting his hands on the right CAR T company. Even when I started talking to the organizations, they wanted the bigger pockets than the bigger entrepreneurs. And I don't blame them. Those took a level of capital I don't know if I would have got to. So you have to be realistic.
And to be really fair, history has not been kind to the second wave of cell therapy companies, right? You know, I think everyone that chased on the tail of Juno and Kite, you know, I don't think there's been much to emerge from that. I think there's been more now that people have become a lot smarter about it, but that second wave didn't fare too well. I'm excited that you did what you did, because quite honestly, in hearing kind of the differences and the advantages of what you do, you're probably in. And just tell me if I'm wrong here. You're probably in an area where you kind of know the things that your peptide should be good at, and your indication selection is probably a little bit more targeted. You probably have a little bit of a better biological thesis about what you can go after, rather than thinking about, you know, we have a platform that can do anything. The whole universe is ours. What should we do?
Yeah, I think peptides really have a great therapeutic opportunity and really are clean in their approach. Like you said, we know what to expect now from all of our molecules. You know, I mean, we ran a phase 1 study with no SAEs or DLTs going up to the top dose level. That, for anyone out there, that means no serious adverse events. This is very well tolerated, these agents, you know, and they're effective. You know, I think those are the kind of therapies we need. We don't need, you know, to destroy the entire body to get to those cells. We need to sort of tip the balance, right? I mean, I think there's a fine comb here where, you know, the body isn't all or none, it's balances. And if we could tip the balance back in the cancer cells from being a pro-survival phenotype, which is how they pick up these enhanced proliferation, growth, travel ability around the bodies, to a pro-death phenotype. And it doesn't mean go from 100 to 0, it means go from 5149 to 5149. We could change the game. So I think using a gentler therapy that, you know, doesn't go after every version of every protein in your body, but gets to those targeted ones you need, is a good future approach here. And that's what we're doing.
Yeah, I think so. So go ahead.
Yeah, I was gonna say, we have specifically on that, we have a 2-part question from our audience. And the first part is specifically about the peptides. How do your peptides get taken up by tumors or across the blood-brain barrier? And do you have specialized cell penetrating tags? And then, so that's more specific around your programs. And then the second question is broader and more sort of finance related. How did you overcome industry's reluctance to look favorably on peptides? Clearly, you've been able to raise a lot of capital. So how did you combine both your vision and the actual overcoming that reluctance?
So I think the first question first. So out of our molecules, I mentioned we keep them to a certain size. Part of the molecule is the active domain that comes from off of the peptide screening system or out of literature or from our license. And the other part is we put in different amino acid sequences that we know allow for cell penetration. And we've characterized over 62 different sequences, and we have an ongoing process to discover our own as well. And we have settled in on sequences that we can use to deliver our peptides to the cytoplasm or to the nucleus. But this is not a conjugation. This is a single peptide chain. Just when we're doing that building, the first set would be the amino acids that would allow for penetration, and the second set would be, you know, the ones that do the activity domain. We have different peptides, like I mentioned, different cell penetrating domains. Our lead peptide, ST101, uses our nuclear targeter. That's been disclosed, so I could talk about it. That is our own derivative from what's called penetratin, which is one of the most well-characterized of cell penetrating peptides. It's derived from Drosophila. And we made some tweaks to it, a couple of amino acid substitutions. That is what's called a cationic amphipathic cell-penetrating peptide. So it has hydrophobic and hydrophilic regions to it, and it's positively charged. So what that allows it to do is passively enter cells rapidly. So it approaches the cell membrane and forms what's called a reverse micelle, where the cell membrane, which is bilayer, sort of inverts into itself. The peptide comes in and it injects it in. There's a video on our website that sort of shows that in cartoon nature, but it's actually pretty accurate to the way it's believed to happen. And then from there, it doesn't get compartmentalized. It shuttles directly to the nucleus. We show within 5 minutes, we have complete cytoplasmic and nuclear penetration. Okay? So the second question, and crossing the blood-brain barrier, we believe it's likely that the penetrating domains help with that aspect as well. And we have a lot of data to quantitate the crossing of the blood-brain barrier for our peptides. For the second question, I'm not sure I have turned industries on that yet. Certain industry folks have, and data really is what tells the story.
Yeah.
We were very fortunate. When I was at that table in July of 2015 at Columbia, Sue Bang and Sammy Tadros from Celgene were at the table with me. And they liked the program as well, but they knew that it was too early for Celgene. So they said if we raised money and formed the company, they would get involved. And they did. You know, they came in with it took me 6-7 months of doing work to get from that October point to that April, May closing of the financing. But they did come in and have been supporters of ours. And then, 5 years later during COVID, BMS had already acquired them. It was a new round of diligence starting from scratch. I think being the Celgene legacy kept their attention during COVID, and they loved everything they saw, and they came on as a shareholder as well. And we have a board observer from BMS who's been wonderful to work with. And since then, I think the data sets have turned the tides with industry. I think that's what's really doing it there. I think with the larger investment funds, I still think it's still an uphill battle to a certain extent. We're getting a lot of traction, a lot of interest, but I think that's where we still have to convince people.
Yeah, and that's where over time your data works. You had to have those early believers, but then you build on the backs of that to get
I think the industry voice ends up being an important one around the table because, quite honestly, those are the people that are going to get the calls on diligence stuff anyway, right? There's the background calls that say, "What do you guys really think of this asset?" and that kind of stuff. And if you have people that are around the table and believe it and understand the story and understand the data and can speak intelligently about it, that's incredibly helpful in a lot of ways. So. I don't know if I think the data probably speaks the loudest, but being able to have that, additional industry support, even if it isn't something where you already have a transaction done, I think that's important.
Yeah, absolutely.
So we had another question about, sorry, I'm being accosted here. We had another question about biomarkers in the thing. I'm sorry, down you get.
Yeah, so the question was basically, look, when you're looking at your business model, right? So how important are biomarkers? I know we talk a lot about biomarkers, and we have for the last several years. And then when in your process do you select what you're looking for?
Biomarkers are, of course, very important. Even more so to industry and investors than they are to the company. We might be able to say we can do it, in a different fashion, and they still want to see biomarkers to know that you're going to be able to, sort of the LOXO model, right? How do we build up a patient enrichment to make sure that we have a specific drug? How to make sure the right people are getting it? Right. I think for a lot of our targets, it's obvious. We go after a lot of the targets that are biomarkers. We're going after beta-catenin, June, FOXP3, KRAS, c-MYC, right? So I think a lot of our biomarkers are obvious, but I think it's important for us internally, right, to develop our programs as we're getting peptides and we're developing from hits to leads. We develop reporter assays that show we could block the changes. Then we go into different cancer types and make sure we get the right genetic changes. And we always identify what the right biomarkers and downstream targets will be early on. And we can carry those into the clinic, whether it's through immunohistochemistry analysis, foundation medicine panels, nanostring analysis. So I think it's pretty critical these days to know what you want to look for and how to enrich those patients appropriately.
Yeah. And at the same time, the biomarkers that you're talking about are also, basically a way to explain the biology, right? It's the next level beyond survival, beyond SAEs. and actually getting people comfortable with, why are you seeing what you're seeing? And I think that's pretty important, especially when you have something where you are going into either difficult tumor types or anything along those lines. It probably helps a lot from that, and in addition to any of the actual data angles on the clinical trial. So we can talk a little bit. We have about 15 minutes left. I think we could probably revert a little bit to the ecosystem stuff and how New York has changed. I mean, you mentioned early on about how you always wanted to help build companies up in New York. As you've mentioned, you've been in New York your entire life, and so how is it that you think the ecosystem has evolved, say from the time you came back for grad school to now?
I mean, I think I could almost think about them in what that first decade was like. That 2000 to 2010 decade was filled with, initially, actually, I don't think much promise per se. I think we were very academic at the beginning of the decade here. When I would talk to folks about maybe leaving to go to business school, Scheinberg was exceedingly supportive. A lot of other folks weren't, right? It was still, industry was not a New York science thing. It was Boston and San Francisco was okay, but in New York, we're academic in nature. I think, I went to a talk over at Rockefeller, which was Joel Marcus and Mike Bloomberg, sometime in the mid-2000 timeframe, talking about the concept of the Alexandria Center. And that's the first time I saw that New York was going to make an effort. And I think that happening right at the end of that first decade kicked off 2010 to 2020 for us, in which we saw from Alexandria through biolabs, through J Labs, and now, I mean, there's New York Bio Inc. at New York Med, where we are up in Westchester, stuff on Long Island. I mean, I think since then the amount of lab space available for young startups has exploded. The number of companies in that time frame has exploded. Some bigger companies and successes have happened. A lot of big financings. The coming of Arch and Versant and setting up shop here in the city, Deerfield expanding, right? I think it's been the 2010 to 20 timeframe has set the stage for our explosive growth in the next decade, when our companies start to graduate out of the incubators into 10 to 20,000 square foot spaces, maybe filling out an ecosystem in the suburbs, up Westchester, out on Long Island, getting clusters there, raising more money, getting clinical successes. And I think we have that momentum now where it's not going to stop. Whereas, early on we weren't sure how it was going to go. So I think this is the decade that New York really puts themselves on the map.
Yeah, I would agree. And actually, I think your initial point is an underappreciated one, right? So we have, as we said, I think we have the largest population of postdocs and graduate students in the world in New York City. And it's largely seen as a scientific asset. Everyone loves to hire out of this pool now. But as you said before, 20 years ago, it was frowned upon for anybody to leave academia, right? So when you don't, I think one of the perhaps underappreciated elements of the stuff that we have here is that community of postdocs and graduate students is actually really energized around entrepreneurship, around startups, around new ventures. And 20 years ago, it really wasn't, right? It was really kind of frowned upon to do anything else. So I think that's a really big point that other people probably haven't brought up quite so much because they haven't seen it quite so much.
Derek, I've literally had professors who gave me a hard time in 2000, 2005 timeframe. About 5 years ago, come back to me about starting their own companies and asking for some advice.
It's funny how that works, right?
And it was wonderful. Not in like a karma kind. It was just wonderful to be able to give back to those folks. The business knowledge they imparted so much science knowledge over here that to help them out.
Do you think?
Remember that thing I told you not to do? How do I do that?
I was gonna say, do you think that was more structural within the academic, just sort of the academic lifestyle, that was their path and they had decided that? Or do you think now that more academic institutions are sort of not willing, but they're facilitating the out licensing of their joint IP with the investigators and the scientists? What do you think caused that change within New York, within the New York ecosystem?
Well, I think we have some amazing tech transfer offices here. I think Oren runs an amazing shop, and I just use the word amazing too many times, but I think Oren does a great job at Columbia, and is a model for how we should run tech transfer organizations. I think Sloan Kettering and Cornell, and everyone has these great programs. And I also think that, venture has turned largely to be able to start companies with lots of money to allow professors to pursue what the ultimate, maybe dream of their science is that they can't necessarily do in the academic setting. And like, I don't know, it hurts companies like mine sometimes, I feel like, because we did it on our own. We bootstrapped it, we raised retail money, and really were efficient. But, for professors with great ideas to potentially be able to sort of privatize the late stage academic research into a vehicle where they don't have to worry about money, it's really appealing. And I think
it is that pendulum swinging towards more kind of venture created companies that really kind of favor the academic scientists in a way, because they don't change a lot about what they do, and it takes away a little bit of the I'll call it the initial CEO risk, right? You have a CEO that has to go out and sell this deal to investors, and quite honestly, one of the biggest pieces of risk an investor look at is person risk. So, a great program with the wrong CEO behind it, tends to not get traction and probably doesn't go where they want it to go. And it's been, I think, helpful to put more money behind a lot of science. I'm wondering how much that changes now, right? I'm wondering if there is kind of a need for earlier leadership or more folks to find, assets the way that you did, because really the number of opportunities that get that kind of money isn't very large, and the breadth of science that's being pursued, it is really, really broad. So I'm wondering if there's another kind of creeping of the market back towards, either that type of financing where you have an early stage entrepreneur, or kind of smaller rounds, individual licenses, that kind of thing, that even a company like Sapience would be able to take up. Like when you said you were hunting for new assets earlier.
Yeah, but look, financing a company is all about financial strategy from day one up through year 10. You can't really think more than 5 years at a time, but you have to think 5 years at a time. And that, as I've learned in my career, the quality of each financing really helps with the next financings. You always, so starting with the right money is very helpful. So hopefully, we do see some more early stage, regular size round investments from companies that can get behind companies that don't need 100 million, would be ruined by 100 million, right? But can use a really good 20 million. I think that's what's hard to find,
On the bright side, I think in addition to the number of companies we're seeing form go up, anecdotally, we are seeing more and more folks establish offices here for funds, right? So I think there are kind of new players in that venture ecosystem. I think there are new people that are seeing what's going on here and want to participate in it. So again, I'm very much with you on the next decade of growth for New York, both in New York City and beyond. Right. So actually, I'm interested in this. So can you talk a little bit about being in Westchester and the ability to build a company in Westchester and outside of New York City? Because again, I think Westchester is a great place to build a company, and in some ways it's underappreciated because it's so close to New York City that it gets absorbed a little bit in the news. But talk a little bit about being in Westchester.
I think Westchester is a great landing spot for graduating from New York City. It's a place to lay roots down as a company, and as your company matures and you bring on more staff and more people that have, families and stuff, it's a great place to be for that. Proximity wise, right? You can draw from Connecticut, New Jersey, even Pennsylvania, which is far, Long Island, and the city, right? And from northern Westchester or Rockland. So it's a great spot to draw everyone from easily. Academic university wise, we have great hospitals up here. We have universities, we have biotech incubators. Major company, we have Regeneron, right? We have that foundation company up here. We have Contrafect, we have Accorda, we have other companies up here. We have incubators with 7, 8, 9, 10 companies in it. And what we're doing at the county level here is investing in building out more spaces. So companies like ours will graduate into those 10 to 20,000 square foot spaces and out of the incubators. And as each one of us grows and moves on to bigger spaces, it leaves behind spaces for others. We have a very welcoming sort of Westchester County Biotech Task Force and Biotech Initiative. From George Latimer on down, who's the county executive, all the way up to Albany. We have a great support system here that is investing in the industry here. So I think it's the right place for us to be. For me personally, when you get to pick where the company is and you live in Westchester, I get to drive 10 minutes to work every day, going away from New York City in the right direction. And it's perfect for me personally. But it's been great for our employees too.
That's a nice commute and certainly enviable. And it sounds like you've been able to get back in the lab through the last 2 years and everything. It sounds like you handled that really, really, really well.
So one thing, I'm sorry, we mentioned the lab. I meant to say that, and I'd be remiss. The other thing about Westchester that we had, which was very beneficial to us, is a very big cost advantage, right? When I was looking at spaces, whether it was J Labs or Alexandria, what without talking about prices, what I might have to pay for a 3 or 4 foot lab bench, I can get an 800 square foot lab, right? So I never been able to do what I did and scale appropriately in the city that I was able to do in Westchester. I mean, we have over 1,200 square foot of lab space, right? That would probably be 2 lab benches in J Lab.
Yep.
Yeah. Well, and as New York Bio, we're thrilled with development all across the state. And you're looking at Westchester outside my window. So it's easy for us to root for the development and the expansion, right? And I really do think that we're seeing growth all across the state from the commitment, both that private investors as well as government is making, right? And the economic development incentives that they provide, which it takes all of these things, right, together to catalyze the real growth that we need. And Sapience is an amazing example of a company that you had amazing ideas and you put the talent together, and then the timing was also right? So for your growth in New York, and we're really happy that you're here.
Thank you.
Yeah, I think one of the advantages that New York has is we have a number of spaces that are being developed that are going to accommodate a wide range of companies, right? We've got, we've talked over and over again about the substrate this here. It's really pretty unmatched, and having places that we can go. Quite honestly, I can be from the middle of the city to your labs faster than you can go from faster than you could ever go from Kendall Square over to Longwood, right? And it's people ask where the next Kendall Square is, and I don't know that physically exists, but what I do know is that we have a lot of spaces in and around New York and not too far away that can be successful, which I think is important. So I'm really excited about the kind of companies that are developing here and the places they can go. And certainly yours is something I've been excited about for a long time. I'm psyched that we could have you here on the 6th anniversary of the founding of the company. That's super exciting that the date worked out that way. So why don't you take like the last little bit to kind of wrap things up and let us know where you're going with Sapience next.
No, we're very excited. It's been a great year. We started our first clinical study in the summer of 2020, and we're about to transition to phase 2 clinical studies in November. We're very fortunate that the first patient to receive a therapeutic dose of ST-101 in any of our targeted indications had a clinical response. We've had a very successful phase 1 study with a number of patients with long-term stable disease and responses and very safe drugs. So we're very excited about our future here. We can't wait to get into phase 2, get into those glioblastoma patients, get back into more melanoma patients, breast cancer patients, prostate cancer patients, and have that second drug, move towards the clinic next year. So we're really looking to continue our growth the same way we said that, I think this decade is going to be good for biotech in New York. I think it's going to be great for Sapience as well.
Well, we're thrilled to be a partner with you as a member of New York Bio. And we're especially grateful to you, Barry, for spending time with us and the audience today. So thanks. And everybody have a good day.
Thanks, everybody. Have a good one.
Bye.
Thank you for tuning in to New York Bio's virtual breakfast series. Join us every Tuesday at 9 a.m. for more discussions with leaders from across the healthcare spectrum. For more information on New York Bio, please visit us at www.newyorkbio.org.